All right, here we go. Lipid structure and function. So our goals by the end of this video, or the end of this week, whatever we're working on, you should be able to identify the different types of lipid molecules. If I show you a molecule, you'll be able to say, oh, that's a saturated fat, that's an unsaturated fat, that's a steroid. So identify it based on its structure, describe its structure, know the differences between them, as well as be able to describe their function and explain what is the role of each of these different type of lipids. Okay, so here we go. I have three types of lipids that we're going to talk about. We're going to talk first about fats and triglycerides, their main purpose being storage of energy. Phospholipids are the main component of membranes, and then steroids. Steroids, one of their main purposes is for cell signaling. Okay. So lipids, unlike some of the other molecules we're talking about, carbohydrates, nucleic acids, proteins, lipids are very different in their structures. But we group them all together because they're all very hydrophobic. Remember, hydrophobic being that they don't interact well with water. They are non-polar molecules, okay? And here's kind of an overview of the different molecules that we're going to be looking at. This is a steroid, this is a phospholipid, this is a triglyceride, a fat. Remember, when we're looking at these organic molecules, these skeletal drawings are basically showing us carbon and hydrogen bonds. And if there's anything besides a carbon and hydrogen, it's written out, right? So carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. Most of the molecules here, most of the atoms in this molecule are carbon, hydrogen, right? And remember that carbon-hydrogen bond is non-polar, which means they share the electrons equally, so therefore there's no partial charges. When there's no partial charges to the molecule, or it's not an ion, water can't interact with it, so it's hydrophobic. Okay, nonpolar carbon-hydrogen bonds, hydrophobic molecules, and it's these characteristics that all of these molecules share, which is why we group lipids together into one kind of group of biomolecules. So our first type of lipid we're going to talk about, triglycerides, also called fats most commonly. These molecules are built by kind of two main subunits. We have a glycerol backbone. Glycerol is basically just a 3-carbon carbohydrate. Okay, so 1, 2, 3 carbons. And through dehydration synthesis, we attach on fatty acid chains. So long chains of carbon-hydrogen with a carboxyl group right here on one end. These are usually even in number, 16, 18, 20, something like that, right? As far as the number of carbons during the fatty acid chain, they tend to be even numbered. So 1, 2, 3 fatty acid chains attached to the 1, 2, 3 hydroxyl groups of the glycerol through dehydration synthesis. Right? Dehydration synthesis, hydrogen and a hydroxyl group creates a water molecule. Okay? So when it's all said and done, a triglyceride has this glycerol backbone to it with 1, 2, 3 fatty acid chains coming off of it. Okay? And because the majority of this molecule is composed of these fatty acid chains, which are carbon-hydrogen, carbon-hydrogen, that's very nonpolar, so this is very hydrophobic. So what are triglycerides used for? So triglycerides are used most often for storage of energy. So if we consume excess calories, excess food, and our body doesn't need it for energy right then, we can convert the food that we consume, like carbohydrates or amino acids or whatever, we can usually convert it into fat. Most often we'll convert carbohydrates into fat. And why we do that is because we might have a scenario later on where we might have a lot of energy. And so we can store excess energy as fat molecules. Most often we see fat in animal products because animals don't have a lot of space to store excess energy, right? We're limited in our space as opposed to like a plant, which can grow and take up as much space as it needs, which doesn't need to have that compact form of energy storage, right? So we'll convert excess energy into fat to store later. And fat has way more energy in it per gram than a carbohydrate does, right? So carbohydrate has about 4 calories per gram, whereas fat has about 9 calories per gram. Okay, so more than double the amount of energy stored. So calories is a measure of energy. About double the amount of energy stored in fat than there is in carbohydrates. Okay, and also fats are much more compact. Again, they're much smaller of a molecule than something like a glycogen molecule or a starch molecule. They're much smaller, so it doesn't take as much room to store a large amount of energy in a very relatively small amount of space. And so we can then tap into that energy source later if we need to, for in a scenario where, again, food is not prevalent. Let's say maybe it's been a while since we've eaten. We can use those fat molecules to create ATP, energy for our cells, if necessary. Fats have a secondary purpose of insulation and protection. So we have, like, under our skin, there's a layer of fat to help us insulate and prevent heat loss, right? Most of our internal organs are kind of layered in a small, are kind of wrapped in a small layer of fat, okay? So it's kind of like a lubricant to keep everything, so that way when we move, we're not completely squishing all of our organs together, and to help us insulate, to help minimize any heat loss to the environment, which helps us save energy as well. But those are some secondary purposes to fat. Its main purpose is for storage of energy. Now let's talk about some types of triglycerides. We're going to focus mostly on the difference between unsaturated and saturated fat. If we look at like a back of a food label or something, you'll probably see or under carbohydrates, I think one of like the first thing that's listed is fats, right? So you'll see unsaturated fat and saturated fat, and you might see trans fat as well, okay? But what do those terms mean? Unsaturated fats are fats that have one or more double bonds in their fatty acid tails. It means that they are not saturated with hydrogens because of that double bond, okay? So if we were to count the number of hydrogens on like this tail right here, it would have more hydrogens on this tail, because they're all a single bond, than we would see on this tail right here. Okay, see how this fatty acid tail has that double bond right here? That means that there's fewer hydrogens because of this double bond. Okay, so they're not saturated with hydrogens, that's what that means. And because of that double bond, that causes that fatty acid to bend. Okay, so see, there's a little bit more space between the tails here than there is between these two saturated tails, right? So there's more space here, and it causes this molecule to kind of bend out into space, okay? And so this bend that's caused by this double bond means that these molecules cannot compact tightly together. So they can't compact tightly together, which means unsaturated fats tend to remain liquid, especially at room temperature, because of this double bond, because they can't stack nicely together to form a solid. We might have more than one double bond. We might have multiple double bonds, and depending on where they occur, that can give these fats very different and specific names. I just want us to know, for us, that unsaturated means that there's a double bond, which causes a bend in the fat tail, and that they're not saturated with hydrogens, and that they tend to be liquid at room temperature. So these types of fats, unsaturated fats, are the ones that tend to be most commonly found in plants. They're the quote-unquote healthy fats. So things like olive oil or any oil that's derived from like plants, nuts, seeds, things like that would be an unsaturated fat, or like omega-3 oils would be an unsaturated fat too, okay? So they tend to be liquid at room temperature because of that double bond, because they're not saturated with hydrogens. Compare that to our saturated fat. Okay, so look, saturated fat, this carbon-hydrogen tail, this carbon-hydrogen fatty acid, all single bonds, which means it has the max amount of hydrogens that these carbons can be bonded to. Okay, and look how nice and rectangular shape this is. Okay, so we could easily stack a lot of these together to create a nice, tightly packed in solid. Okay, so when there's only single bonds in the fatty acid tails, that's going to be a saturated fat. Okay, these are the types of fats that tend to be solid at room temperature. Okay, so often we'll find these in animal products. So we tend to find them in things like butter or in meat that animals produce, right? Dairy as well tends to have some saturated fat in it as well. Some plants will produce saturated fat. Things like coconut oil is a saturated fat. Okay, so coconut oil is usually solid at room temperature. There are some health concerns with consuming large amounts of saturated fat, but for us, again, the main difference that I want us to worry about is being able to identify this is a glycerol molecule, these are fatty acid tails, and being able to look at it and see, are there double bonds here in these tails? No. Okay, so that means it's a saturated fat. One other fat that you may hear about or see on like your food labels or is a trans fat. So let's talk about what trans fat means and why it comes up in the news somewhat often, right? So when we're talking about double bonds, we have two conformations of those double bonds. We have what's called the cis and the trans conformation, right? So we've used these terms in our everyday life before, probably. Cis and trans, right? So cis meaning same, right? And trans meaning across. So if we look at this cis fatty acid, if we're looking specifically at that double bond, what is on the same side here of this double bond? These hydrogens. Okay, that's what we mean by cis double bond. So these two hydrogens are on the same side compared to these carbons. Okay, they're both on this upper end right here. Trans means across. So hydrogen on the top over here, but for this other carbon that's involved with the double bond, the hydrogen's on the bottom. Okay. And so this conformation doesn't have as much of a bend to it. The trans conformation doesn't have as much of a bend to that molecule as the cis conformation does. And what that means is now that this trans fatty acid is pretty solid compared to this cis fatty acid. Okay, it's going to be more solid at room temperature. So most naturally occurring unsaturated fats are going to be in the cis fatty acid. There are some naturally occurring trans fatty acid, most commonly found in things like red meat or dairy, but most naturally occurring unsaturated fats are going to be in the cis conformation where the hydrogen's on the same side. But what we do, one thing that we figured out how to do is we take these unsaturated fats from vegetable oil, things like corn oil, canola oil, things like that, and we do what's called hydrogenation. Basically, we add a bunch of hydrogens to them, and that changes the conformation of this bond during hydrogenation from cis to trans and turns it into a solid, like margarine, right? Hydrogenated vegetable oil is basically, we take this and we turn it cis into trans, which makes it a solid. But why do we care, right? Why does this come up in the news? So trans fats, or consuming diets that are high in trans fats, has been linked to stress in our endoplasmic reticulum, which is an organelle in our cell that we'll learn about soon, that helps with processing toxic substances. It also helps with producing proteins and carbohydrates, things like that. High amounts of trans fat in diets has been linked to increase of fat storage in the liver, and we all know fatty liver disease is not good, right? Too much fat in the liver can't function properly. It also, high amounts of trans fat seems to promote cholesterol synthesis, which can also have huge major impacts on our cardiovascular health, right? And overall, there's just a positive association with consuming with a diet that's high in trans fat and cardiovascular disease, okay? And one of those impacts could be blockage of arteries due to potentially high levels of cholesterol, okay? Less blood that's flowing could have higher blood pressure. If it's an artery that's in the heart, that could lead to a heart attack, right? There's a whole bunch of health concerns associated with trans fat, which is why the FDA has started to regulate trans fat in foods because of the major health implications. But you can have up to, like, I think 0.8 grams of trans fat in a serving, and nutrition label still label that as 0 grams. Okay, so it's not a perfect system, but there are some steps that are being taken to regulate trans fat. Okay, this video is getting pretty long, so we're going to say this is lipids part 1, and we'll start on part 2 with phospholipids in our next video.